So let's quickly talk about the degradation of nucleoproteins. Degradation of nucleoproteins. Now, the exam question was like this. Using a schematic representation, describe or discuss the degradation of what? Nucleoprotein. Okay. So the most important thing here was for you to actually have a diagram. Now, this is a very popular diagram in my medical school. And for the particular diagram in my medical school, all these spaces like this, they actually have their enzymes, or will I say catalysts. So please, if you are watching this video and actually from my medical school, you have to go back, right? Go back and look at the native diagram that we have in school that have the various enzymes and catalysts. I think this one is it detergent that is supposed to be here or something. All right. So we'll use this image to actually what discuss what degradation of nucleoprotein is. Right. So nucleoproteins are actually what a complex. They are complexes of nucleic acid and proteins. All right, complex of nucleic acid and proteins. Do you know what that means? They are made up of proteins. So for the protein, there's no need for breakdown. But nucleic acid is also still made up of different components. So that's why the nucleic acid had to be broken down up to the level of this. You get? So there are complexes of nucleic acid and proteins, and they can undergo degradation through various cellular processes. Okay. So what are the mechanisms of nucleoprotein degradation? I think this is a better image, closer to what we have in school. All right. So for the mechanism now, yes, you actually find a better diagram for degradation of nucleoproteins. You can even grab this one. This one is close to being better. Okay. So now once you do that diagram, then you now add one or two notes. So the notes is basically on the mechanism, all right? So you have enzymatic degradation. So you now say that specific enzymes actually break down these nucleoproteins. For example, RNases will break down RNA components. DNases will break down DNA components, right? Then there's proteasomal degradation. So where you say that the proteasome is a complex. Okay, sorry. The proteasome is a protein complex that degrades or damage. Okay, that degrades, damage, or unneeded proteins. Okay, including those in proteasomes. Okay, so need be it that this protein is broken down, what will act on it is the proteasome. All right. So there's lysosomal degradation too. So lysosome enzymes, okay, so the lysosome contains enzymes that break down nucleoproteins into their what constituent nucleic acid and amino acids. All right, that's just by the way. Then autophagy, this process involves degradation of cellular components, including nucleoproteins within the lysosomes. Right. So what is the importance of this degradation of nucleoproteins? Degradation of nucleoproteins is crucial for maintaining what? Cellular homeostasis, recycling nucleotides, and regulating gene expression. So defects in these processes can lead to what? Various diseases, including what? Genetic disorders and cancers, right? So the most important thing is a diagram for this question. Now for the diagram, you can just grab one or two things and say about it. Or if not, you can just draw the diagram, then start describing it from the diagram. You just say nucleoproteins broken down to nucleic acid and proteins by the aid of what? Is it gastric acid or pepsin or detergent, right? Then the nucleic acid is further broken down into nucleotide by the aid of what? RNAs or DNAs. Do you understand? So you just describe the diagram from the own drawing. Okay, so either way you answer this, I think you'll be on the good side. Right, so that's it about what? Discussion of the degradation of nucleoproteins.